Welcome to a new tutorial presented by DSA Charles AB. The tutorial is from Atlas of Neural Anatomy, Frank H. Nater, John Criggs, and James Perkins. Neural Anatomy. In this tutorial, we're going to see the optic nerve. Now, to visualize the optic nerve, the first question we need to ask ourselves is, number one, is the optic nerve a sensory or motor nerve? It's a sensory nerve. It's a spatial sensory nerve, and characteristics of spatial sensory nerve is that spatial sensory nerve are bipolar. So the bipolar neurons have their nuclei, have their cell bodies lying outside the central nervous system, and this is why the optic nerve is going to have at first a ganglion called the geniculate ganglion, which is shown here. This is the geniculate ganglion here. So the geniculate bodies, geniculate ganglion, as it is shown also at the level of, so this is the lateral geniculate bodies below, which is below the thalamus. So now, in order to visualize the optic nerve, we need to know that we need to first know the constituent at the level of the retina. So for the optic nerve, the first point starts at the periphery. So from this point here to this last point here. So we have these cells here. These first cells here are the R cells and the C cells. R cells are the rods, and we have the cones. These are the two main cells which are involved with the removal of different colors, with the images involved at the level of what you are seeing. So the rod cells are much more specific cells which are capable of removing all the colors. We have the different rod cells. We have the cone cells are the cells which are much more specific. So they are used to remove the different colors from the eyes. So the two main colors of the eyes which are involved are the red, blue, and green, and all of them are capable of being removed by the cone cells. Now, and the cones are much more found. The location where the cones are in high concentration are at the level of the macula densa. So what happens is that the eye is always maintained with all its processes such that the image is always lying at the level of the macula densa for a high precision. Now the rod cells are these cells here, and these cells here are much more they are not much more specific, but they are capable of visualizing as small light as possible. So now with the cone cells, you are not capable of seeing in the dark with the cone cells. In the dark, we instead visualize with the rod cells, because they have a high capability of taking as small impulses as possible. So as all these cells are going to be activated, they are going to send it to these other cells here. We have these H cells here, the horizontal cells. So these are the horizontal cells here. They are going to send now to the B cells. So these are the bipolar cells here. So now as they are connected, we see that one rod, as we can see here, one cone is connected to one bipolar cells directly, but the rod is connected to one bipolar cell is connected to many rods compared to the cones. So you see that the body takes much more consideration of the cone cells than the rod cells. So one rod, three rods here are connected to one bipolar cells, while only one is connected to one bipolar cell. So as it goes now into the brain, so you have these horizontal cells which are also used in order to inhibit the bipolar cells, as you have also seen with the olfactory neurons. So it's the horizontal cells used to inhibit in order to prevent the colors of one location, the position of the imaging. So in order to make sure that the position of the imaging is effective. So we also have this other cell here, A cells, are called the amacrine cells. So they are also used in the inhibition of the lateral cells here. So the bipolar cells are going to give now to these other cells here, and these other cells are going to be called the ganglion cells. So we have the G cells here, the ganglion cells, and the ganglion cells are going to give their tracts which are going to move now into the central nervous system and go to the lateral geniculate body, which is found at the level of the thalamus, the inferior part of the thalamus. So now what do we have to note for the optic nerve? at first we need to know the different region that the optic nerve are going to take. So we need to know that the lateral optic nerve, which are located here, are taking images from the nasal region, while this optic nerve here, which is the major optic nerve, are going to take images from the temporal region. So these images are from the temporal region here. The temporal region starts from this portion and is going to end at this portion here, while this lateral one, which is taken from nasal region, are going to start from here and going to end here. So these are different images that they are going to take. Now, upon the overlapping of the left and the right eye, you're going to have this precise image here. So this is a precise image that the brain is going to consider, and these are the boundaries or the outlet of the image. So this is why when you close the one eye, you realize that the image is always different from the image that is produced when you have the two eyes open, because the positioning is slightly arranged. So this is what you have to note. So now the darker circle represents the macular zone. At this point here, this dark circle is the macular zone, where you are going to have a much more precise location. So at this center here, you have a much more precise location. But what they have also forgotten to put here now is the zone where you are going to have the optic disc. So the dark zone. You also have one zone where you have the dark zone, where you are not capable. It's a dark point. If you visualize just on a clear whiteboard, you're going to see a dark point, a portion of your eye which is where there's no image. It's a small point which is dark. So now this is what you have to visualize. Now this one major part of the eye is taking at the temporal zone, and this one is going to take from the nasal zone. So you have this. So now at the level of the eye, it comes here. So the nasal tracts are going to, the lateral tracts are going to move laterally and going to continue here to the geniculate ganglion, while the nasal tracts, which are taking images from the temporal zone, are going to move now and going to do they are going to decussate at the level of the optic chiasm to come to the contralateral geniculate body. So this is what happens. So if you have a lesion here, you are going to lose your optic nerves, your nasal part of the optic nerve, and the nasal part of the optic nerve take the temporal images. This is why a lesion here causes a bitemporal hemianopsia. You're not going to see at the bitemporal zones. Now, if you have a lesion here, you're going to have a homonymous hemianopsia. This is why all the light which are coming in this particular direction are not going to be seen. So in this direction is going to be nasal view, and this direction is going to be the nasal view. So this is the temporal view, and this is the nasal view. So the light which come in the same direction. That is why the word is say homonymous. So if you cut here, you're going to have a homonymous hemianopsia. The light is coming from the same direction. Similarly here. So now as it takes the contralateral, the nasal fibers goes into this nasal part, the optic nerve, which are in the nasal part, are going to come now and going to bind with this other one here and to form a tract. This is the optic tract here. They are going to enter now into the lateral geniculate body. In the lateral geniculate body, they are going to give the optic radiations, and the optic radiations are going to now go to the occipital lobe. So mostly the portion of the occipital lobe which is involved is the cuneus and level the calcarine sulcus. So the calcarine sulcus is going to divide both of them. So we have projection on the right dorsal geniculate nuclei. This is a projection which is seen on the right dorsal geniculate nuclei, consisting of both this one here. So this one is taken from temporal zone, and this one is also taken. So you have this one projection here. So it gives the optic radiations. So you see that this is a calcarine sulcus. Now the calcarine sulcus, the image which is seen above the calcarine sulcus is this particular image which is located here. Now the image which is seen below the calcarine sulcus is this image here, which was located at this zone here. So below the calcarine sulcus was this image here, above the calcarine sulcus was this image here. So now we see all the images which are homonymous, seen in the same direction. This one here and this one here are taken in this particular direction. So all of them which are homonymous are going to be involved in the formation of image in this left calcarine sulcus, and this portion is going to be right and this portion is going to be left. So it's going to form at the level of the right calcareous sulcus here. So, and this is going to form at the level of the left calcareous sulcus. All the images are also homonymous in this direction. So nasal view and temporal view here are going to be formed here. Now the inferior image is going to be superior in the above the calcareous sulcus, while the superior image is going to be inferior below the calcareous sulcus. So from here we've seen all the different the cause of the optic nerve. So if you have a lesion, you need to know that if you have a lesion at this portion here, you're going to have instead a quadrantanopia. So you're not or you are going to also have a contralateral hemianopsia. So this is either a contralateral hemianopsia. At this portion, you're going to have a contralateral hemianopsia, or you're going to have a quadrantanopia. So from here, visualize all the different causes of the optic nerve with the different lesions that you can have. So thanks for your kind attention, and please don't forget to like and subscribe for Science Video Maker. Thank you.